Bismillahirrahmanirrahim. Assalamualaikum and good morning to the examiners and to the panels who are watching my videos. My name is Nur Shazim binti Karsono, and today I would like to present about my final year project, which is about plant microbe interaction in rice with endophytic Escherichia coli ASFL2. As a beginning, E. coli is known as a harmless microorganism present in our environment. It can be found in lower intestine of warm-blooded organism, as it is a part of normal flora found in a gut. Their presence gives the benefit to the host by providing vitamin and also preventing the establishment of pathogenic bacteria in the intestine. As a consequence from this condition, the population of E. coli in the environment become much more stable in their new habitat. As the abundance of E. coli can be found in the soil, it creates an interaction with the plant. E. coli, also known as one of endophytic bacteria. Endophytic bacteria means that the bacteria that can be found in surface sterilized plant or extracted inside the plant. But the seed is known as one of their main source to maintain their survive within the plant internal tissue. Their presence will not cause harmful effect to the host, and seed is known as one of the sources to maintain their survive in the host. This is due to the high nutrient availability obtained from the host, which is plant. The advantage from this beneficial interaction includes it's capable to promote the plant growth and also increase plant defense system against plant pathogen. This interaction is not visible to the naked eyes, but this interaction occurs in different ways at different levels. This interaction also can be classified as pathogenic, saprophytic, and also beneficial interaction. When this abundant microorganism that we found in the soil started to colonize the rhizoplant. Rhizoplant are the environment in the soil that around the roots of the bacteria that actively colonize the root of the plant is known as plant growth promoting rhizobacteria. But why they started their colonization at the root surface? This is because root is reported to be one of their main site of entry in order to establish the colonization within the plant internal tissue. In addition, plant growth promoting rhizobacteria itself are able to tolerate under stressful conditions, and it's also able to utilize and provide enough nutrient for the plant in soluble form. The problem statement in this experiment is on how these endophytic bacteria are capable to promote the plant growth, and if they can promote the plant growth, how the colonization process occurs. The objective of this experiment are to investigate how the colonization of endophyte occur in rice by using microscopic technique, and also to study the influence of the phytohormone production in enhance the plant growth. There are a few methods used to achieve the goal of this experiment. In this experiment, the bacteria and plant material used are rice seed MR220 and also E. coli USM-L2. The confirmation tests of E. coli USM-L2 have been done before the experiment and also during the experiment. This is to ensure there is no contamination occur during the process. Gram staining technique is used in order to confirm E. coli USM-L2 are gram-positive or gram-negative bacteria. For biochemical test, since the bacteria I used is E. coli USM-L2 and we know it belongs to Enterobacteriaceae, and I decided to use indoor test. Next, we move to the enumeration of bacterial colonization by viable cell count and also phytohormone production assay. For this assay, we need to germinate the rice seed before we inoculate with and without E. coli USM-L2. Before we left the seed for germination process, we must ensure that the seed have been surface sterilized. After 7 days of germination process, we will start to inoculate the rice with and without E. coli USML2. We left the tube for 48 hours and 96 hours. In this experiment, there are two treatments, which is one with rice without E. coli USML2 and another one rice with E. coli USML. After 48 hours and 96 hours, we will start to extract the rice plant. Once we get the extraction, then we will start with spread plate method. Same goes with phytohormone production assay. We need to have the extraction, and once we have the extraction, we will add it with Salkowski reagent. This is known as Salkowski calorimetric technique. 1 ml of extraction will be added with 2 ml of Salkowski reagent, and the test tube will be left for 30 minutes for dark reaction to occur. After 30 minutes, the absorbance reading will be obtained by using spectrophotometry, and the reading will be recorded. For the elongation of plant assay, after 48 hours and 96 hours inoculation, the rice will be taken up and placed into a piece of paper. The rice is divided into two parts, which is aerial and also root parts. Next, in order to know if there any migration occur, so we decided to do transmission electron microscope. The suitable thickness for the observation under transmission electron microscope is estimated between 10 to 20 nanometer. When we cut the sample and we get the gold color, it means that we can take the sample to be observed under transmission electron microscope. Result and discussion. First, for the confirmation test of E. coli USML. Figure 4.1 shown that the observation that have been made under light microscope with 100 magnification. From this observation, we can determine that this bacteria is gram-negative bacteria. Why I'm saying this E. coli USML2 is a gram-negative bacteria? It is because the pink stain appear on the bacterial cell. From this observation also, we can know the morphology of the bacteria. E. coli USML2 is a rod-shaped bacteria, and the arrangement of this bacteria is arranged singly and some are in pairs. Once we have done with the gram staining technique, we proceed the test with biochemical test. For this biochemical test, I decided to use indole test. By adding the correct reagent into the broth, the formation of red layer on the top of the broth can be. This reaction involves deamination and also hydrolysis reaction. It breaks down the amino acid, which is tryptophan, to release indole, with the presence of enzyme known as tryptophanase. Survival cell count. As we can see in figure 4.3, A has the highest mean of colony per unit compared with B. It refers to the growth medium of the rice. While D is higher than F after 48 hours inoculation, this is due to the colonization process that is started from the root surface. After 96 hours of inoculation, the means of colony forming unit increase in all parts of the plant, include the growth media. In phytohormone production, we are focusing on indole 3-acetic acid production, and we tested by using Salkowski calorimetry technique. As we can see in figure 4.4 and 4.5, the concentration of IAA are highest after 96 hours inoculation in aerial part compared with aerial part after 48 hours inoculation. Since we want to investigate the effect of the presence endophytic bacteria for the plant growth, this assay reveal with the presence of E. coli USML2, the concentration of IAA is higher compared the rice without E. coli USML2. IAA is important for development of shoot and also the root, as it's involved in cell elongation. This analysis revealed that it is highly significant. The presence of E. coli USML2, the concentration of IAA is high. It means that the presence of this endophytic bacteria help to regulate the plant hormone and also indirectly will support the growth and development of the plant. Elongation of plant assay is related with the phytohormone production. Since we know IAA concentration is high, will result with increasing in length of the shoot and also the root of the rice plant. The concentration of IAA increased after 96 hours inoculation. The aerial part of the rice also increased compared without E. coli USML. The increasing in cell elongation occur after 48 hours and also 96 hours inoculation with the presence of E. coli USML2 in both root and aerial part of the rice. Concentration of IAA will stimulate the elongation of root tip and also the shoot of the plant. See in this figure, A, B, and C is the rice without the inoculation of E. coli USML2, while the rest has been treated with E. coli USML2. As you can see in figure 4.8D, there are abundance of E. coli USML2 have been found inside the root. It means that the colonization are started from the root part before it started to migrate to other part of the plant. Root attachment of this endophytic bacteria and the colonization play a vital role for efficient invasion. Once these endophytic bacteria are successful invade the plant cell, it will start to colonize the area and start to utilize the nutrient available. There is a migration of E. coli USML2 occur since we can see the presence of E. coli USML2 in the stem and also the leaf of the rice. The presence of plant cell wall degrading enzyme make the colonization process much more successful and they can migrate from the root to the upper part of the rice. The spreading of this endophytic bacteria started from the root until to the leaf of the rice resulted as they are much more stable and they become established within the plant internal tissue. To sum up, this experiment revealed that E. coli USML2 have the capabilities to migrate from the root to the upper part of the plant. The presence of the flagella in E. coli USML2 are the advantage for this endophytic bacteria to migrate within the plant internal tissue. With the presence of this endophytic bacteria, it enhances phytohormone production, which is also act as plant growth regulator. This beneficial interaction that occur between the plant and the microbe increase plant fitness and at the same time help to minimize the application of chemical fertilizer in agriculture activities. For future recommendation, plant microbe interaction must be discovered more on its advantage for agriculture activities. And we also need to know the implication from this interaction in the future. Question 1. How can we confirm the seeds are free from endophyte? The seeds are free from endophyte based on the observation under transmission electron microscope. The seed without treatment show an absence of microorganism compared with the seed treated with E. coli USML2. Second question. In section 3.11.2, explain where do you start measuring the rice. For this section, I divided the rice into two parts, which is aerial and root part. Before taking the measurement, the rice is placed into a piece of paper for better measurement. The upper part of the seed is considered as aerial part while the rest are the root part. For the second part of this question asks on how I reduce the error when measuring. First, I will double check the measurement for high accuracy. Next, I will make sure the instrument I use has highly precision and also the working environment should be controlled to minimize the error when measuring. Question 3. For statistical analysis, justify why I use Duncan for post-hoc analysis and no other tests, such as Turkey. For the statistical analysis, I decided to use Duncan for my analysis as it involves multiple comparison to compare a set of mean, which is help for significant difference determination. That's all for me. Thank you.